Unusual circumstances can preserve exceptional fossils, from insects preserved in exquisite detail in amber to the intricate detail of tissues and cells revealed in coal ball peels, to the soft-bodied creatures of the Burgess Shale, over 500 million years old, preserved on the side of a mountain. Such fossils tell us much about the paleontology and paleoanatomy of past life, details of how bodies were organised and even details of soft parts that don't usually fossilise. But how about how those animals lived, how they moved, how they ate? Did they live as loners, or did they associate in vast herds or shoals? Can we tell anything about their day-to-day life in the same way that a field biologist can about creatures living today? Can we add greater depth to the picture of a fossilised creature beyond skin, bone and muscle? This might seem like an impossible task for the paleontologist. The material we're looking at is, after all, as dead as a doornail, or as dead as my career as an underwear model if you like, but the tools of logic and deduction can help paleontologists act as detectives to piece together the lives of long dead creatures. In previous shows, I have demonstrated how the fossils themselves can provide clues that can be used to bring dead bones and shells back to life. But there is something else too, a powerful class of fossils that I'd like to bring to your attention today, a class of fossils that essentially is fossilised behaviour of organisms. So in this episode, let's ask, what is a trace fossil and how do they form? What do trace fossils tell us about the evolution of life? What traces do creatures leave about how they moved, fed and built a home? And how can fossilised behaviour track changes in environments? These are trace fossils, very recent ones. They show that an animal passed this way not long after the cement was laid. From the print you can tell many things. The gait or stride of the animal, potentially how heavy and how large it was. It also ties this animal to this specific environment and something else about its behaviour too. In this case, a dog's unswerving interest in trotting across cement that you've just spent several hours getting just right. This is also a trace fossil. It shows an insect larva, how it fed on a leaf, its host plant. Not only do we know the name of the host plant from a perceptive botanist, but we can also know the group of insects that produce this mine on the leaf from an equally perceptive entomologist. And if we are truly interested in this interaction, we can determine what tissues this larva preferred to feed on, and in this way, something about the evolutionary history of insects that mine leaves for a living. Trace fossils are found in both marine and terrestrial environments and can be made by a variety of creatures. In sediments, there can be tracks and trails and burrows and borings. In the world of plants and insects, they record plant damage produced by feeding, egg depositing, pollinating, and a whole host of other activities. Ever since life got big, it has been interacting with the environment in a very physical manner. Creatures have been disturbing the physical structures that occur in sediments, like fine laminations or ripple marks, and basically giving things a good mix, what we call bioturbation. The study of trace fossils is called ichnology and can essentially be regarded as the study of fossilised behaviour. Unlike body fossils that consist of the actual parts or impressions of an organism, trace fossils have limited use in biostratigraphy, that's the dividing up and correlation of rocks in a time sense, but trace fossils have several advantages in other areas of paleontology. Firstly, trace fossils often develop under specific environmental conditions, making them great for paleoenvironmental interpretation. Another distinct advantage is that you can be certain that a trail or footprint has not been moved. This is a problem with body fossils if you're trying to use them to interpret an ancient environment, as you have to make sure that the creature has not been moved from the environment to another environment post-mortem. But for a trace fossil, where you find it is where it formed. Secondly, they can also give us an appreciation of the activity of soft-bodied creatures that rarely fossilise. Trace fossils have been studied for a long time, although initially many of these lines and squiggles in rocks were often misidentified as seaweeds or worms. Dinosaur footprints are a little easier to interpret, of course, but were often regarded as footprints of huge flocks of birds. This misidentification of some of these tracks and trails as worms and plants may explain why trace fossils are named like fossil plants and animals, using the Latin binomial system with a genus and a species name. The fact that we are not dealing with an individual species but a type of fossil behaviour, though, can cause some confusion. You just ask some of my long-suffering students. For example, consider this trace fossil. It is given the generic Latin name of Rusophycus. However, as this is a type of behaviour, in this case where an organism rested, Rusophycus can be produced by a whole range of different organisms, by a polychaete worm, a snail, a shrimp, and a trilobite, just to name a selection. Another difference we have to consider relates to the different things organisms may do on a day-to-day basis. Just as I could potentially leave multiple different traces in wet sand by making sandcastles, digging for clams, walking, running, or more commonly in my case, tripping and falling flat on my face, any individual fossil organism could be responsible for an entire range of traces, just depending on what it was doing at the time. Consider this visualization. A single trilobite is shown moving from a resting position where it generates one of those Rusophycus resting traces, to starting to a furrowing behaviour called cruziana, and then picking up its skirts, well actually it's plural flanges, and doing what we call in Britain a runner and scarpering on out of there. In this trace fossil, we can tell based on the orientation of the marks in the cruziana trail that this individual approached from the right side, chewing through the sediment as it moved, and eventually, after a busy time ploughing through the muck, decided to take a break and rest for a while, leaving another Rusophycus, those resting traces. There are, of course, more than just resting, feeding and moving traces. Just see how many traces the common fiddler crab can leave behind, from a living burrow, various types of walking traces, a radiating trace of processed sand that the animal leaves when it's feeding, and poop. Yep, poop is a trace fossil too. This potential confusion when it comes to studying and naming trace fossils is why we tend to classify them by behaviour they represent and not by the creatures that produce them. I would like to give you a brief description of the types of trace by behaviour that can be found. We will look at some specific examples in more detail a little later on. These are repicnia, traces that an animal makes as it moves. The cruziana left behind by a trilobite trail or the footprints of a dinosaur are repicnia. Passicnia are traces left by animals not only moving but grazing at the same time. There is also evidence of what can only be described as paleontological farming, the so-called agricnia. For example, the honeycombed paleodicton that we'll meet later. Phodicnia describe various feeding burrow structures. Domicnia are interpreted as traces where an organism actually lived. For example, a prairie dog tunnel system. And cubicnia covers all types of resting traces like the Rusophycus we described earlier, and this impression of a resting starfish called Asterocytes from the Devonian of northeast Ohio. As you may have realised, the divisions between some of these classifications may not be hard and fast. Perhaps a creature's living burrow could also act as a feeding burrow. It does, however, provide a framework upon which we can start to hang various types of fossil behaviour. As you can see, trace fossils are an important part of a paleontologist's arsenal for interpreting past behaviour and environment, but there is another important component to the study of trace fossils, that is in understanding the early evolution of the biosphere and the diversification of animal life. The first fossil evidence of life is found around 3.4 billion years ago in the fossil microbes found in the Strelley Pool sandstone of Western Australia. In the same section though, laminated structures have been found that have been interpreted as stromatolites. Stromatolites are laminated structures commonly produced by photosynthetic cyanobacteria. These bacteria trap grains of sediment in their sticky mucilaginous sheath and then move upwards through the sediment, creating a new layer. In this way, they commonly dome upwards, producing column-like sedimentary structures. You can see modern examples exposed above water in the Hamlin Pool Marine Nature Reserve in Western Australia. Stromatolites can be classified as trace fossils as it is not the cyanobacteria that are being preserved, just the laminations and the structures they produce. For billions of years, although we find individual, single-celled microfossils, stromatolites are really the only large-scale evidence of life that we have. Their diversity over time also has a story to tell. Throughout the Precambrian, they are a common component of many shallow marine settings, but by the time we get to the Cambrian period, they are only at about 20% of their former abundance. This change in stromatolite abundance marks a critical part of the story of life on Earth. It tells us that grazing organisms are now an important part of the ocean system. Grazers are now feeding on the bacteria on top of the sediment before they get a chance to form the beautiful columns and domes we see in the Precambrian. This change is called the Cambrian substrate revolution, where not only grazing animals were starting to have an impact on the planet, but also burrowing animals too, who would churn the sediment still further. After the evolution of various worms and arthropods during the proliferation of life we call the Cambrian explosion, sediments in shallow marine environments went from fairly firm, stabilised by all those microbial mats, to more, well, mushy. Since the Cambrian, stromatolites have been marginalised, restricted to extreme environments, places where the water may be too salty or too oxygen poor for grazing creatures. Areas like these in Shark Bay, Australia, provide a rare window into the way the world must have looked billions of years ago before the evolution of grazers. Another type of trace fossil, coprolites or fecal pellets, poop, may have also made a significant contribution to the Earth system at this time. By producing large fecal structures that sink and deliver carbon rapidly to the ocean floor and sediments, they lessened the oxygen demand on surface waters. This may have promoted oxygen enrichment in the oceans and allowed for the evolution of larger and even more complex creatures. We should all be thankful for poop. But there is one more interesting item regarding the evolution of trace fossils that I'd like to highlight. Remember I said that trace fossils are pretty useless when it comes to biostratigraphy, that is, timing events in geological history. Generally, geological boundaries are marked by the incoming of a distinctive new fossil species that can be correlated across a wide area. For example, the base of the Carboniferous period is defined by the appearance of a conodont microfossil, Siphomophila sulcata, in a geological section in Brittany in northwestern France. There is one point in geological time, though, probably one of the most famous and important points in the history of the planet, where it is a trace fossil and not a body fossil that are used to define the base of a geological period. But not only a period, for the trace fossil assemblage Treptochinus pedum not only defines the base of the Cambrian period, but also the whole Phanerozoic eon, marking a transition from the Precambrian into a world full of complex life. Geological periods are defined at sections called international stratotype sections, places where rocks are used to define a particular boundary in time within geological history. For the base of the Cambrian, the transition from the Precambrian to the Cambrian and the entire Phanerozoic eon is taken at the place called Fortune Head, Newfoundland. It is here that the appearance of Treptochinus and an association of other trace fossils defines the base of the Cambrian. It is thought that the creatures producing this strange trace fossil was a creature called a priapulid worm, probing in the sediment, preying upon or scavenging for small invertebrates just in or on the surface of the sediment. The significance of this assemblage is profound though. It marks a critical event in the biosphere in which organisms are now starting to dynamically interact with the physical environment, an evolutionary change in the behaviour of life and the biosphere. Let's focus in on some of that advanced behaviour now. let's see how trace fossils provide insights into how life leaves a record of some pretty fundamental behaviour, specifically how creatures have moved, how they fed, and how some of them built a home. A mobile fauna is a big leap forward, if you'll pardon the pun, for the biosphere. As we saw earlier, by the time we get into the Cambrian, various creatures, like trilobites, are engaged in a variety of different activities, from furrowing through sediments to picking up their skirts and scurrying across it. But not all marine creatures simply meander around the surface of the ocean floor. Sometimes movement is needed in another direction when a catastrophe occurs. Well, a catastrophe if you're a marine invertebrate. I'm thinking here of a class of trace fossils called Forgychnia, or escape tracers. One such trace, Diplocriterion, is a U-shaped burrow that shows curved laminations or spretae on the inside and outside of its U-shaped burrow. The outer spretae form as the organism moves rapidly, adjusting its position upwards in response to a pile of sediment being very rapidly dumped upon it. It would be wrong to assume that all trace fossils are found in marine deposits, though. And to illustrate this, here is a movement trace probably created by an insect like a bristletail, insects closely related to silverfish that were walking and sometimes jumping across a tidal flat during the Lower Permian about 270 million years ago in southern New Mexico. After the initial description of these unique traces and trails in 1997, the same type of traces have now been found in several other localities worldwide. Now we know what to look for. These traces are turning up everywhere. Of course, we can't talk about movement traces without mentioning dinosaur tracks. We have to be careful when interpreting these, though. Just from this example here, a bunch of questions present themselves. For example, were these tracks made all at once or at different times? If at different times, how soon after? Seconds, minutes, hours, days? Also, how many individuals? We obviously have 2 species of print, but was it just 2 individuals or many? But we can glean some very important information about such tracks. For example, if you had a set of prints, you can determine stride length, the distance between one footfall and the next footfall. Also, when we shift from a walk and into a run, stride length will increase. A British zoologist, Robert McNeill Alexander, saw an opportunity here when he noticed a relationship between stride length and an animal's speed, providing you know the distance from the hip to the ground. He also noted that this relationship was true whether the animal was a quadruped, like my friend Jacob here, or a biped, like an emu, human, or of course, a 2-legged dinosaur. He developed a formula that expressed this relationship, where u is the velocity, g is the acceleration of free fall due to gravity, d is the stride length, and h, the height of the hip from the ground surface. The whole thing is simplified to this. There is an obvious problem here, though. Because we're looking at a trace fossil, the individual who produced the trace is often unknown. In the case of dinosaurs, fortunately, we have an out, as there is a predictable relationship between hip height and foot length for each dinosaur group. Hip height is found to be about 4 to 6 times foot length, depending on what group of dinosaurs you're dealing with. From this, we think that the walking speed for most dinosaurs is about 1 to 4 meters per second. That's similar to humans, with running speeds around 10 to 15 meters per second for some of the smaller carnivorous bipedal dinosaurs. The fastest dinosaur is clocked at 54 kilometers per hour. That's about 35 mph, similar to a fast racehorse today. Trace fossils have also helped answer questions about how marine vertebrates move too. Consider the Triassic semi-aquatic marine reptile, Nothosaurus. A long paleontological debate has centred around how these animals swam. Did they swim with their limbs in a kind of a figure of 8 motion? This is the mode of locomotion penguins use today. Or did they employ more of a kind of a rowing motion with their limbs? Paleontologists from the University of Bristol and the Geological Survey in China found a series of pairs of slot-like tracks preserved in mudstones from Yunnan in southern China. Analysis of the orientation and size of these traces indicate that they were made by the animal's forelimbs as they moved over the seafloor using a rowing motion in unison, not a figure-of-8 motion. But there's more too. These fossils also provide insight into some of the hunting behaviour of these animals. Some traces have been interpreted as foraging trails. Apparently Nothosaurus was using its forelimbs to disturb the mud on the ocean floor and then snap at fish or crustaceans as they try to swim away. All this biomechanical and behavioural information about a long extinct animal provided by marks and gouges in sedimentary rock. And of course, our branch of the biosphere has been leaving footprints in paleontological wet cement for many millions of years as well. Some of the first, and probably some of the most famous, are the Leteli footprints in Tanzania, discovered by Mary Leakey in 3.6 million year old volcanic ash during her 1976 field season. The prints were probably made by Australopithecus afarensis, the same species as the famous Lucy fossil. These footfalls record humans walking through ash made wet by rainfall. As we have noted, many things can be deduced from a footprint. These show that our early ancestors walked by the heel of the foot first, followed by the toes pushing off at the end of the stride, the so-called heel strike toe off walking, very similar to the way that we walk today. Other footprints belonging to our ancestors are being studied at the Smithsonian. Footprints of Homo erectus in Illoret, Kenya, dating to 1.51 to 1.53 million years ago, have been found showing how humans at this time were really starting to walk very much like we do today. Researchers such as National Museum of Natural History scientist Dr. Richard Potts and Kay Behrens-Meyer are hoping that footprints will be found at this site that will provide even more insight into the evolution of human walking and running too. So, ever since the biosphere has started purposely moving around, it has been generating trace fossils. But complex life doesn't just move, it also has to eat, and in doing so produces a whole additional type of trace fossil. We have already mentioned one of the strangest types earlier, Paleodicton, a honeycomb-shaped structure found in quiet deep water environments from the Cambrian to the present day. This particular type of trace is called an agrycnia, a farming trace, and has been interpreted as organisms deliberately cultivating bacteria on the ocean floor. But what could possibly be producing such regular geometric patterns? The deepwater submersible DSV Alvin went searching for modern Paleodicton near volcanic vents that lie about 3,500 metres underwater along the mid-Atlantic ridge. They found and took samples of structures very similar to Paleodicton, but unfortunately could not find the creature that produced them. The mystery continues. But probably the most common feeding traces are those made by deposit feeders, animals that process sediment for organic material and nutrients. There are many different types, but two common ones are Rhizocorallium and Zoophycus. Rhizocorallium is generally found as burrows that are shallowly inclined to the sediment bedding planes. They can be over a metre long, most are smaller, but the animal that produced these structures is still a mystery, one of the many that keeps paleobiology such a fascinating and exciting discipline. Zoophycus, by contrast, produces a more vertical spiral trace as it moves through the sediment searching for goodies. Of course, not all feeding traces are vertical. Some just wander over the surface of the sediment. These particular sediment gulpers will generally produce traces that have no overlapping parts, a good strategy if you want to avoid processing the same area twice. But everyone needs a place to call home. Dwelling traces, or domicnia in trace fossil terminology, can be simple vertical burrows like Scollithus here, or more complex ones like Ophiomorpha, a trace decorated with sediment balls that the trace maker stuck on the outside of the burrow. These are horizontal branching dwelling traces called thalassinoides found in Jurassic limestone in southern Israel. Such burrows can be generated by a number of creatures like fish and crustaceans, but specifically by Thalassinoidea, after which this particular type of trace is named. Recently, some unexpected dwelling traces have come to light in both western Montana and Victoria of Australia that show that some dinosaurs dug burrows. Unusually, we actually find the remains of the creature producing these structures in the bottom of the burrows themselves. This is Eryctodromeus, meaning digging runner, a genus of fairly small, about 6.8 feet long, ornithopod dinosaur from the mid-Cretaceous. The burrows in Montana are about 2 meters long and 20 centimeters wide. The possibility of vertebrate burrowing behavior can likely be pushed back even further than this though. In the Triassic rocks of Argentina's Ichigualasto Basin, a series of large burrows dating to around 230 million years ago have been discovered by paleontologist Karina Columbi and colleagues. It is thought that at least one type of these burrows was produced by cynodonts, a type of reptile that were closely related to the early mammals. As useful as trace fossils are in paleobiology, they are also extremely useful in paleoenvironmental analysis too. Trace fossil assemblages, or ichnofacies as they're known, respond rapidly to environmental conditions in which they form. During the 1960s, the utility of trace fossils in paleoenvironmental interpretation really took off. Pioneering work by researchers such as German paleontologist Adolf Dolf Seilacker and later Robert Frey have really expanded our appreciation of trace fossils in this regard. It's interesting that the general character of these trace fossil ichnofacies has remained fairly constant from the Cambrian to the present day. The implications of this is that although the producers of the traces have changed through time, the manner in which they were responding to their environment, their behaviour, has not changed very much at all. Or put it another way, it's a bit like a play that's been running for hundreds of millions of years, but the script has largely stayed the same, but the actors occupying the roles has changed many, many times. The ichnofacies are named by reference to one particular characteristic trace fossil of each assemblage. Let's look at a very small selection from the deep to shallow water. The Nereides ichnofacies is characterised by horizontal, meandering or spiral feeding traces, and sometimes the bacterial farming traces that we mentioned earlier. Vertical burrows aren't very common. This assemblage is indicative of very quiet, low-energy water conditions typically found in deeper oceanic settings at a distance from shore. The Cruziana ichnofacies is a very busy trace fossil assemblage, full of feeding, moving, living and resting traces. This facies is usually characterised by sands and silts deposited in shallow marine settings with a high biodiversity. The Scollithos ichnofacies is our shallowest facies and is dominated by vertical burrows and is usually associated with coarser sandy material, like we'd find close to shore in the surf zone. Here, shifting sediment on the ocean makes living on the surface of the substrate difficult. This explains why creatures that burrow vertically tend to dominate. You can see how tempting it would be to link different ichnofacies to water depth, and there is a bit of a relationship, but probably a more useful factor to consider would be water turbidity. Consider, for example, the case of the Cardium Formation, a rock formation of the late Cretaceous of Alberta in Canada. The sediments demonstrate an alteration in conditions between quiet and more turbid water. During quiet times, the sediments are dominated by sands and silts and are characterised by typical Cruziana ichnofacies. The traces are mostly produced by mobile carnivores and deposit feeders, chewing through or ploughing over the fine-grained nutrient and organic-rich sediment. This relatively quiet environment is occasionally interrupted, though, by coarser, clean wash sands that contain a typical vertical-orientated Scollithos assemblage, demonstrating many of those escape Fugignea burrows that we described earlier. These clean sands have been interpreted as storm events, with sediment being washed back from nearshore environments into deeper waters by storm, surge, ebb currents. During these events, the quieter water Cruziana fauna would either be killed or displaced and replaced by the high-energy Scollithos fauna. When the storm surge currents waned and normal sedimentation resumed, the Cruziana fauna returned, so trace fossil assemblages not only tie down specific environments, they can also help chart changes in environmental conditions over time, whether that is long-term change, as you might expect with variations in sea level, or just short-term events, like the ones we've just examined from the Cardium Formation. All of these sediment-based trace fossils, as well as evidence for the interactions between plants and arthropods, are an extremely valuable tool that we can use to understand the behaviour of extinct organisms. They also chart critical environmental changes in a way that body fossils sometimes cannot provide. We've barely scratched the surface of the study of ichnology and plant-arthropod interactions. Hopefully, though, you can appreciate what a vital part fossil behaviour in all of its variety has to play in the interpretation of our planet's history.